Okay, can somebody confirm you can hear me?
Yes.
Yeah.
I can hear you.
Thank you. Is my volume coming in good? Let me turn that up a little.
Yeah.
Yes, it sounds good.
Good. All right, let's see where we are. So today is May 14th. And let me share my screen here. Should be able to see my screen now. So today is May 14th. We should be starting chapter 6. I don't think we're there yet. I think we need to finish chapter 5. There will be a lab today, lab 8. I've got that we were talking about the summary of aerobic respiration, anaerobic respiration, and fermentation. Anyone disagree? All right. Lipid and protein catabolism can be done as well as glucose and obviously carbohydrate metabolism. So we can use lipids and protein for food and then burn it in aerobic respiration. And microbes can also do this. There is a problem, however, with lipid and protein catabolism, and that is lipids and proteins are too big of molecules to come across the cell membrane. And how that's solved, both for ourselves as well as microbes, is we or they secrete extracellular enzymes and then digest these large molecules outside the cell, breaking down the protein, for example, into amino acids. And then the amino acids are brought across the cell membrane into the cell and then can be metabolized in the normal way, or at least they can be metabolized for aerobic respiration. Usually with amino acids, they will be converted into glucose and then sent down aerobic respiration in the normal way. Although cells can burn directly lipids and amino acids, or I should say the derivatives of lipids and amino acids. And I'll show you a little bit about that in a little bit. So lipases and proteases are secreted outside the cell, and they will break down the lipids and proteins into their monomers. For lipids, the monomers will be glycerol and fatty acids. The glycerol will be catabolized in glycolysis, meaning glycerol and fatty acids will be brought into the cell. Glycerol will be catabolized by glycolysis. The fatty acids will be cleaved into 2 carbon chunks and then sent through the Krebs cycle. Amino acids, the amino group will be removed. And then depending on which amino acid it is, some of them can be sent in glycolysis, others can be sent in various stages of the Krebs cycle. And this is trying to show you the interrelationship of carbohydrate, lipid, and protein metabolism. Let me blow this up. So lipids broken into glycerol and fatty acid. Glycerol sent in midway through glycolysis. Fatty acids broken into 2 carbon chunks. They'll be converted into acetyl coenzyme A and then can enter the Krebs cycle. Proteins, depending on which amino acid we are, some of them can be entering glycolysis, some can be broken into 2-carbon chunks and then converted into acetyl coenzyme A. Others enter late in the Krebs cycle. Most carbohydrates are broken down into sugars. The sugars are converted into glucose, and then glucose enters aerobic respiration in the normal way. Any question about any of that? All right. So metabolic pathways use energy, and we can create energy and store it as ATP by catabolizing carbohydrates, lipids, and proteins. Let me back up just a minute. We can burn lipids directly in aerobic respiration, but a marathon runner, for example, will not burn lipids in aerobic respiration until 45 minutes in their marathon run. The first 45 minutes of their marathon run, they will be burning carbohydrates and obtaining ATP from carbohydrate catabolism. But after that, after running for 45 minutes, a marathon runner can burn lipids in aerobic respiration. I don't know about protein, and it may depend on which amino acid we're talking about, how long it takes. But once again, proteins do not immediately enter aerobic respiration. We will burn carbohydrates preferentially in aerobic respiration to make ATP. The oxidation of glucose in aerobic respiration is the most efficient for generating ATP. It is much more efficient than fermentation. And even the best forms of anaerobic respiration are not as efficient as aerobic respiration. The ATP that's generated is used for various cellular processes, including transport of materials, the movement of cells or organisms, the synthesis of new cellular components like anabolism. Most cellular ATP is used for anabolism, making up larger molecules, including the biosynthesis of protein, carbohydrates, and lipids. Any question about any of that? I suppose we could also make nucleic acids, and that makes all the 4 molecules of life. Now, metabolism is not totally separate. There is an integration of metabolism where anabolic and catabolic pathways are joined by common intermediates. We call these amphibolic pathways if the metabolic pathway may function in both directions, meaning if catabolism can occur and yet the same reaction anabolism can occur, this is an amphibolic pathway. Amphibolic pathways bridge the pathways involved in carbohydrate, lipid, protein, and nucleic acid metabolism. And I think you know this. If you eat too many carbohydrates, you will initially start storing it down in your liver as glycogen. However, if you continue eating carbohydrates, you will then convert that carbohydrate into a lipid and then store it down as a lipid. I think everybody knows that. And as I've already mentioned, if you were to eat a high fat or high protein diet, so you don't have enough carbohydrates to send through aerobic respiration, you can burn lipids and protein and then send it through aerobic respiration. The important part is amphibolic pathways bridge the pathways in carbohydrate, lipid, protein, and nucleic acid metabolism. Any questions about that? So this is trying to show you the integration of metabolism. Wherever we have a double arrow, that means this step can go this way in the Krebs cycle, but it could also go this way in an amphibolic pathway. And you'll notice that in aerobic respiration, almost everywhere we have a double arrow. And then this right here, as well as this right here, it has the double arrow this way, meaning I don't know why they've chosen to draw it this way, but you can go this way in aerobic respiration, but you can also go from this molecule to this molecule by going this way. And that's why that arrow is that way. Any question about that? And then the same with the lipids. You all know that if you were to eat too much carbohydrates, come on, you will store that as a lipid, meaning the carbohydrates are broken down to here and then to here, and then they're converted into fatty acids and glycerol, and then they will join to make the lipids. Any question about any of that? And that's true for the proteins over here, although it doesn't show making the protein, it just has the amino acids. But the lipids is shown in its complexity. And then for those of us who are couch potatoes, you will convert your glucose or carbohydrate into glucose and then break it down this way into acetyl coenzyme A and whatever that is, dihydroxyacetone phosphate, to convert it into glycerol and convert it into fatty acids and make the lipids. And those couch potatoes, when we eat a lot of lipids, you won't send the lipids directly into aerobic respiration, breaking it down into glycerol and breaking it down to fatty acids. Instead, what we'll do, us couch potatoes, is we will break down the fatty acids into acetyl coenzyme A, and then acetyl coenzyme A and glycerol will be converted into glucose, and then we'll send glucose down aerobic respiration in the normal way. Now, obviously, somebody who's a marathon runner or an Olympic athlete or somebody like that, meaning a real athlete, can burn lipids directly in aerobic respiration. But like I say, even a marathon runner, they won't do that until 45 minutes into their marathon run. Any questions about any of that? And let me tell you that this is showing you pretty much the integration of metabolism in humans, as well as microbes and green plants for aerobic respiration. But green plants are much better at amphibolic pathways. They have many more of them than we do. Any questions about any of that? All right, if not, let's move on to photosynthesis. Photosynthesis, as you know, is the conversion of light energy into chemical energy. The chemical energy, like the making of ATP and NADPH, can be used to convert atmospheric CO2 into glucose, and that we call carbon fixation. Photosynthesis is performed by cyanobacteria, algae, and then obviously green plants. So there are many bacteria which can engage in photosynthesis. And then algae, and that makes a lot of microbes. Photosynthesis is a mechanism for synthesizing organic compounds from CO2 and water. And then this allows animals and other microbes to feed on the matter that the photosynthetic organisms have made. Life would not be possible for animals without the photosynthetic organisms. Photosynthesis only occurs in photosynthetic organisms. These organisms contain a light-trapping pigment. One of them, you know, is chlorophyll. Light releases electrons from chlorophyll, and then the energy from the electrons, which initially came from the light energy, is converted to chemical energy via an electron transport chain. That energy is used to make ATP and or NADPH. And oftentimes ATP and NADPH are used up by the photosynthetic cell to form glucose from CO2 and water. And the making of glucose from CO2 and water is called carbon fixation. Any questions about any of that? All right, here's something you probably don't know. There are two types of photosynthesis: cyclic photosynthesis and non-cyclic photosynthesis. Non-cyclic photosynthesis is probably the photosynthesis you were taught in biology 60. I should shut down my email. I've got to open up the email to log into Canvas, and so I've got the email open. That's what that ringing was. In non-cyclic photosynthesis, there is actually two types: making of oxygen, or oxygenic photosynthesis, and then anaerobic photosynthesis. We're going to skip anaerobic photosynthesis, but in both of them, you convert 6 CO2 and then 12 other molecules. In oxygenic photosynthesis, that's 12 water molecules in the presence of light to generate one glucose, C6H12O6. And then in oxygenic photosynthesis, you generate 6 oxygen molecules and then 6 water molecules. All of these oxygens come from the water. None of these oxygens come from the oxygen and CO2. All of this is used to make glucose in the water. Any question about any of that? Like I said, you should know that there is a anaerobic photosynthesis that does not make oxygen, but you don't need to know the equation. So you really don't. I'm not going to quiz you on that. And then there's cyclic photosynthesis, where glucose is not made. What is made in cyclic photosynthesis is ATP. In cyclic photosynthesis, all that's made is ATP from ADP and phosphate. And all photosynthetic organisms that I know of, certainly green plants and cyanobacteria, can engage in either non-cyclic photosynthesis or cyclic photosynthesis. And which one they will do will depend on which molecule they need. If they need ATP, they will engage in cyclic photosynthesis. If they need glucose, they will engage in non-cyclic photosynthesis. Any question about any of that? All right, you should know the chemical equation for non-cyclic photosynthesis as well as in cyclic photosynthesis. Cyclic photosynthesis, called cyclic photophosphorylation, because you actually phosphorylate ADP to make ATP. Let me blow this up. Once again, uses light. The light hits the chlorophyll, which makes 2 electrons leave the chlorophyll. The electrons then enter an electron transport chain. When the electrons leave the chlorophyll, they'll first go into that protein of a electron transport chain, meaning a chain of proteins in the membrane. And then the electrons will move to this protein. And that will give this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. Then the electrons will move to that protein. And then the electrons will move back into this protein, which is photosystem 1, and the electrons will end up where they started, and that's the chlorophyll molecule. And that's why we call this cyclic photosynthesis, because the electrons move in a circle, ending up, come on, mouse, ending up where they started from. Cyclic photosynthesis. Any question about any of that? All right. The hydrogen ions build up on this side of the membrane, so there will be more hydrogen on this side, as well as more more plus charges on this side than that side. So these hydrogen ions will want to move to this side of the membrane by the laws of diffusion. And they can only move through this membrane at this protein, which is an ATP synthase enzyme. And when the hydrogen ions move through this protein, they give the energy this protein needs to phosphorylate ADP, meaning combine ADP and phosphate to make ATP. And that is how ATP is made in cyclic photophosphorylation. Now, this should be familiar to you because this electron transport chain is very similar to the electron transport chain of aerobic respiration. Let me see if I can find it here. There it is there. The difference is it's a different electron transport chain, but it does the same concept, and that is electrons are brought to the electrons in a chain in a membrane, and some of these proteins can pump hydrogen ions from this side of the membrane to that side. The hydrogen ions are then used to make ATP as they move through this ATP synthase protein. Where this happens in a eukaryote this is the chloroplast. So this is the inner membrane of the chloroplast. And this would be inside the chloroplast. This will be between the inner membrane, which will be right here, and then the outer membrane, which will be here. In a bacteria cell, this electron transport chain is inside the cell membrane. This will be the cytoplasm. This will be the cell wall. And the bacteria will pump the hydrogen ions into the cell wall. Any question about any of that? All right. So just a summary of ATP production. The energy source can vary. It can be glucose, or it can be carbohydrate, or for that matter, lipids or proteins. The electrons will be stripped off of glucose or whatever molecule we're digesting. And then the electrons will largely be carried by an electron carrier, which we talked about for aerobic respiration. It'll be NAD plus and FAD. But the energy source can be the sun. The energy source can also be an element, like elemental sulfur or ammonia. And this will be the chemosynthetic bacteria, stripping the electrons from a molecule like an element of sulfur, and then carrying those electrons and electron carriers, sending the electrons to an electron transport chain to make ATP, shown here. This would actually be more likely glycolysis, that ATP. The electron carrier can vary, like I said, in photosynthesis. It's not NAD plus, it's a molecule similar, NADP plus. And then the electron acceptor can vary. Let me see if I can shrink this. Oh, that's a little too much. Well, we'll just end the show. Blow it up that way. The final electron acceptor can vary. For aerobic respiration, it will be oxygen as the final electron acceptor. For anaerobic respiration, it will be a molecule other than oxygen. It could be nitrite, could be nitrate, could be sulfate. For fermentation, it will be an organic compound. It's not shown here. I don't know why it's not, but the final electron acceptor for cyclic photosynthesis is chlorophyll, the molecule that the electron first started from. Any questions about that? The point is that the electron donor molecule can vary and the electron acceptor molecule can vary in the making of ATP. Any questions about chapter 5? If not, let's begin chapter 6. Oh, I should mention that I did some grading, and let me stop sharing my screen. I think it was lab 3. I got graded for the second time. And then the last lab, 6 or 7, I got graded for the first time. And for that lab, if you didn't get 100%, you can make corrections by this Saturday. Looks like I didn't finish all of worksheet lab 3. But lab 7 is graded for the first time, and I'll try and put a note on that lab that you can make corrections by this Saturday for full credit. Any question about any of that? I also got the last worksheet in the lecture, lecture 5, graded. Oh, and I think I got graded 1 or 2 of the extra credit studies. Yeah, extra credit study 3 and extra credit study 4 were graded. Any question about any of the grading? I meant to talk about that in the start of the class and I just forgot. All right, let's begin chapter 6. There we go. Let me see if I'm sharing my screen. I'm not. Let me share my screen. Chapter 6, microbial growth. This slide is showing you the major goals and a rough outline Always know the terms. 2, understand the various requirements of cells for growth. And because we're talking about microbes, you could also say for organisms for growth. 3, understand the phases of growth. There are 4 different phases. And then 4, compare the different methods for measuring microbial growth. Any questions about what we're going to cover? Microbial growth, when we're talking about microbes, when we talk about growth, we are referring to an increase in the number of cells, not an increase in the cell size. I used to teach middle school students, and when we talked about them growing, we were not talking about them increasing in number. Any question about any of that?
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about that? There are various requirements for microbial growth. And if you don't have these requirements, the microbes cannot grow. There are physical requirements and chemical requirements. Physical requirements include that the microbe must be at the correct temperature, the correct pH, and the correct osmotic pressure, meaning must be in the correct salinity. Chemical requirements include the organisms must have a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus, a source of trace elements, a source of oxygen if they require oxygen, and if they require the absence of oxygen, then they require the absence of oxygen, and organic growth factors if they require them. Any question about that? Each species has its own temperature requirements, where a species of microbe can grow at a minimum temperature or a maximum temperature, and below the minimum or above the maximum, the microbe cannot grow. And then the microbe also has an optimum temperature where it will grow the fastest. Microbes can be classified into groups on the basis of the preferred temperature range. So we have microbes that we call psychrophiles, and they grow best around 10 degrees Celsius. That's refrigeration temperature. How can I blow this up? There we go. There are psychotrophs, and these organisms grow best around room temperature. These would be organisms growing in the soil or on your floor, something like that. Mesophiles are organisms that grow best around 37 degrees Celsius. That's 98.6 degrees Fahrenheit. All human pathogens are mesophiles, so you should know that. Thermophiles are organisms that grow best around 62 degrees Celsius. And then hypothermophiles, these are organisms that were not even known when I took microbiology. These organisms grow best around 95 degrees Celsius. Remember, at 100 degrees Celsius, we have water boiling. Any questions about any of that? Now, that's the optimum. Each organism also has a range for what its minimum and maximum will be. The mesophile, something like 10 degrees Celsius, which is refrigeration temperature, to 50 degrees Celsius. Many mesophiles will not grow at 10 degrees. like E. coli will not grow at 10 degrees. The ranges as well as the optimums are not rigidly defined. For example, organisms that grow on our skin, like Micrococcus luteus or Staphylococcus epidermidis, they do not grow best at 37 degrees Celsius. They actually grow best at 30 degrees Celsius, meaning 30 degrees Celsius is their optimum growth temperature. Yet we still call them mesophiles because they're close to a mesophile, or what the majority of mesophiles are, 37 degrees. Any question about that? And then obviously the range will vary with each species. For this one here, something like 10 degrees to 50 degrees. Like I said, E. coli, I don't know what the minimum temperature would be for E. coli, but it's something more like 15 degrees Celsius. And I don't know what the maximum is for E. coli. The point is, the exact characteristics is not rigidly defined. Any question about any of that? No, I shrink that a little bit. Food spoilage temperatures. When food is kept between about 15 degrees Celsius to 50 degrees Celsius, the microbes can grow rapidly. And after 4 hours at this temperature, you should probably throw that food away. Microbes can grow slower at other temperatures, like between 52 degrees to 62 degrees, or between, let me blow that up, between about 15 degrees Celsius down to about 5 degrees Celsius. Many bacteria can survive and some may grow. And this is actually refrigeration temperature, at least below 10 degrees. And so microbes can grow in your refrigerator. And that can spoil the food too. And that's why if you have food without preservatives in it, after the food has been in the refrigerator for about a week, you should probably think about throwing that food away because microbes can grow at that temperature. And even if they're slowly growing microbes that live best at this temperature, so that they won't cause an infection in you, the meaning they can't survive in you because you're too warm, they can cause the food to spoil. And that's why meat and, I don't know, sweetbreads or something like that should be thrown away after about a week in the refrigerator. Now, if the food has preservatives in it, such as high salt concentration or high acid or high sugar, like ketchup has both high salt, high acid, it can survive in the refrigerator much longer. In fact, I have a bottle of ketchup, which is at least from 2018, and I'm still using it and I'm not dying. And that's because ketchup has so many preservatives in it, high salt, high acid, that when it's in the refrigerator, it can last a long time. Pickles is another thing which lasts a long, long time. Almost any of the condiments, like mustard. I think mustard has both vinegar and high salt in it. Any question about any of that? Food can spoil if it is in the danger zone between about 15 degrees to 42 degrees Celsius for too long. What happens is that the microbes can grow, and even though they will not be visible and the food will taste fine and look fine, the microbe can produce a toxin after only, how many hours is that? For about 4 hours. And the real problem is that if you put the food in the refrigerator in too large of a batch, or you put too much of it in the refrigerator all close together, then it won't cool quick enough and the microbes can grow and then produce the toxin, and that'll make the food toxic. What you want to do is separate the food into smaller batches, and then the food will go through the danger zone from about 42 degrees to 15 degrees in an hour or so, and then we'll be at cool enough temperature after that that the microbes cannot grow and produce the toxin. Any question about any of that? All right, if not, let's move on to the physical requirement of pH. Most bacteria grow best around a pH 7, so something like pH 6 to 8. In lab cultures, this is actually a problem because as the microbes grow, they often can produce acids, and the acids will inhibit their growth. To minimize this, like when we make media, we often include a pH buffer in the media so that the microbes can then grow in the media. And then when they make the acid, the buffer will neutralize it so the microbes can continue growing. Molds and yeast actually can grow best at a slightly acidic environment. So they don't like pH 7 the best, although they can grow at that. They prefer between pH 5 and 6, which is slightly acidic. Acidophiles, on the other hand, grow best in acid environments, like a volcanic environment, where the pH will be between pH 0 and pH equal to 3. Or Helicobacter pylori, which grows in your stomach, which can get down to pH 1.5, and it grows best in an acid environment. So down to 1.5, maybe even down to pH 1. And it grows best something like pH 3 to pH 1. Any question about any of that? Oops, didn't mean to be there. Osmotic pressure is another physical requirement. Microbes require a certain amount of water. And you can remove that water by putting in high salt, such as salted meat or molasses, high sugar, or salted fish. High osmotic pressure can remove the necessary water from a cell, like when you put a cell in a hypertonic environment, higher solute concentration outside the cell than inside the cell, shown here, where water will then leave the cell membrane will pull away from the cell wall and then shrink and shrivel, and that can lead to plasmolysis, which generally will inhibit microbial growth, and that will kill some cells. Like when E. coli falls into a bottle of molasses, that E. coli cell will die by plasmolysis from the high osmotic pressure of the molasses pulling out of the water from the E. coli. Any question about any of that? We have used high osmotic pressure to help preserve food for centuries, probably for millennia, such as drying meat or drying fruit. The Asians dry vegetables. I don't think the Westerners dry vegetables, although we do it for preserving. Actually, we do dry beans, for example. And then you can cook the dried beans. Other examples, salted meats, salted fish. molasses. Any question about any of that? Halophiles are organisms that prefer growing in a high salt concentration. There are two types of halophiles, an obligate halophile and a facultative halophile. An obligate halophile requires high salt concentrations in order to grow, like the organisms growing in the Dead Sea between Jordan and Palestine tend to be obligate halophiles. And I can't think of one off the top of my head, but if you take them out of the high salt concentration, those cells will die. A facultative halophile does not require the high salt concentration to grow, but can tolerate salt at greater than 2%. Some can grow in 5% salt. And a facultative halophile that all of you actually know are all of the Staphylococcus family, like it's not Micrococcus, Staphylococcus aureus is a facultative halophile. It can grow on your skin, which can get as high as 20% sodium chloride. But it can grow in a general purpose media like nutrient agar or TSA, which is fairly close to osmotic pressure, meaning about, isotonic solution, which is about 0.9% sodium chloride. Any question about any of that? In a hypotonic solution, one with low osmotic pressure, meaning there's lots of distilled water or water around, water can enter the cell and then kill the cell by osmosis. If the cell doesn't have a cell wall, water will enter the cell and the cell membrane will expand. More water can then enter the cell and the cell membrane will expand until it bursts. And the cell will burst by low osmotic pressure. If the cell, on the other hand, has an intact cell wall, the cell membrane can only expand up until the size of the cell wall. And then the cell wall will protect the cell membrane from expanding any further. If the cell membrane cannot expand, more water cannot enter the cell. And that's how the cell wall can protect the cell. Organisms have a carbon requirement. Carbon is typically half of a cell's dry weight. So if you get any cell and simply remove all the water, half of the remaining weight will be carbon. Carbon is used in structural organic molecules. It's used as an energy source, like, as you know, the carbon in glucose is used to make ATP. Heterotrophs use organic carbon sources. Autotrophs can use CO2 dissolved in the air or in the water. So autotrophs use their carbon source from CO2. Nitrogen, sulfur, and phosphorus are other chemical requirements of microbes. They must have a source of nitrogen, a source of sulfur, and a source of phosphorus in order for the microbe to grow. And let me state that's true of all living things. You have to have a source of nitrogen, sulfur, and phosphorus. Nitrogen's about 14% of the dry weight. I'll probably never ever quiz you on that, but you should know that nitrogen is in amino acids, in proteins, in DNA, in RNA, in ATP. So nitrogen's in a lot of the biological molecules. Nitrogen can be obtained from the decomposition of proteins. And that is actually where humans get their nitrogen from. Many bacteria can get their nitrogen from decomposition of proteins like E. coli, but many bacteria can get their nitrogen from some other source like ammonium or nitrate. As you know, green plants can use nitrate. That's their favorite source of nitrogen. And that's true for many green photosynthetic bacteria as well. A few bacteria can even get nitrogen from the atmosphere and then fix nitrogen into the ecosystem, pulling it down from the atmosphere and then making that nitrogen available to other organisms in the ecosystem. Usually that happens when they die. The bacteria that pull the nitrogen down, the nitrogen will then become available once that bacteria dies. Any question about any of that? Sulfur can be found in amino acids like cysteine, methionine. It's also found in thiamine and biotin, 2 B vitamins. And sulfur is actually used in DNA and RNA synthesis of some microbes. Sulfur can be obtained from the decomposition of proteins, and that's the main source that we get sulfur from. But many bacteria can use sulfate as well as green plants. But some bacteria can use sulfur in H2S, what is that, hydrogen sulfide, which is a toxic gas to us. And some can even use elemental sulfur as their sulfur source. Any question about that? I think most bacteria can probably use sulfate. Those that can't use sulfate either use another sulfur source or can get it from digesting protein. As you know, humans cannot use sulfate. And obviously hydrogen sulfide gas is a toxin to us. Any question about any of that? Phosphorus is another chemical requirement for organisms. Phosphorus is found in DNA, in RNA, ATP, and then phosphorus is found in phospholipids of the cell membrane. So phosphate is an important source of phosphorus, and to my knowledge, all organisms can use phosphate. Certainly humans and most microbes. Any question about any of that? All right. Trace elements are required by all living things. Trace elements tend to be inorganic elements required by microbes or other cells in trace amounts. Why it's called a trace element, meaning they're things like copper and iron. So they actually are an element. And they're often used as enzyme cofactors. Examples of trace elements include iron, copper, and zinc. Usually, most microbes, as well as people, can get trace elements from their environment. Like we get our trace elements from the food we eat and the liquid we drink. We don't need to take any vitamins or whatnot to supply the trace elements because usually there's enough trace elements in the food we eat and the liquids we drink. Occasionally there will be an exception, like in Western Oregon and Western Washington, because of all the rain has washed iodine out of the soil. And iodine is a trace element. Many people in the world simply get it from the food they eat and the liquid they drink. In Western Oregon and Western Washington, we would not get enough iodine from the food that we grow in Western Oregon and Western Washington, as well as the liquids we make in Western Oregon or Western Washington. Now, obviously, a lot of the food we get now is grown from elsewhere, like almost all the beef in the store. It's grown from out of state and then brought to the supermarket. So you can get it. But in the old days, it was more of a problem. And what the US government did, because there were a few places like obviously Western Oregon and Western Washington that were lacking in iodine, they put iodine in salt. And so if you got iodinized salt, you had a source of iodine. Any question about any of that? Organic growth factors are biological molecules that the organism cannot make itself. It needs to obtain this organic growth factor from its environment. Examples of an organic growth factor for humans would be vitamins, like vitamin C is an organic growth factor. We must get this from the food or the liquids we drink because we cannot make vitamin C. Certain essential amino acids and essential fatty acids are another organic growth factor. Like for us, the amino acid lysine is an organic growth factor. We cannot make that amino acid. We can't convert another amino acid into lysine. So we must get lysine in the food we eat or the liquids we consume. Purines and pyrimidines can also be organic growth factors. We'll talk more about them when we talk about nucleic acids. Any question about any of this? All right. Oxygen is another chemical requirement. For aerobes, they absolutely must get oxygen or else they'll die. For an anaerobe, many anaerobes must have the absence of oxygen or they will die. Obviously, some organism can tolerate oxygen. Some organisms can even use oxygen, but they do not require it, like a facultative anaerobe can use oxygen to make ATP, but it doesn't require it. They'll fall back on some other means for making ATP than aerobic respiration if oxygen is not around. Oxygen is, in a sense, a toxic substance. Even to aerobes, oxygen can be toxic because there are certain metabolites of oxygen which are just frankly toxic. Any question about any of that? So yes, we are an aerobe. We must be around oxygen or we'll die. But there are some forms of oxygen, some metabolites of oxygen, which are toxic to us too. And our cells have to have some way of dealing with that. There are 4 toxic forms of oxygen. Oops, let me get out of that. Come on. There's singlet oxygen. This is oxygen boosted to a high energy state. This oxygen is extremely reactive and it will bump into other things and then damage the other thing. It's actually present, singlet oxygen, in our phagocytic white blood cells, and the white blood cell can use singlet oxygen to kill what the white blood cell is trying to kill, meaning the white blood cell can come up to something and then release singlet oxygen on that something, and the singlet oxygen can then do damage to that something. Potentially, if you get enough of that damage, it can kill the thing the white blood cell is trying to kill. Any questions about any of that? Superoxide free radical is another form of toxic oxygen. Let me blow this up. We have it as a single symbol O2 with that, I don't know what you want to call that, open circle or square, and then a negative charge. This refers to an unpaired electron, only found in free radicals. What this superoxide free radical will do is bump into another molecule and then steal an electron from that other molecule, damaging that other molecule. Superoxide free radicals are actually made in aerobic respiration. And then these molecules can do damage in the cell by bumping into another molecule and then stealing the electron from the molecule, damaging the molecule. How we neutralize superoxide free radicals is we have an enzyme, superoxide dismutase or SOD, which can take two superoxide free radicals and two hydrogen ions to make hydrogen peroxide and oxygen. Oxygen itself is not toxic to our cells, but hydrogen peroxide is. And so if SOD makes hydrogen peroxide, the cell then has to have a means for handling this peroxide, H2O2. And that brings us to our third toxic form of oxygen, peroxide anions, like H2O2. Hydrogen peroxide is a peroxide anion. This also is toxic to cells. What we do to neutralize peroxide anions is we have an enzyme called catalase, which takes two peroxide anions, in this case, two hydrogen peroxide, and then converts the hydrogen peroxide into water and oxygen. And catalase is an enzyme in our cells. Any question about that? Most microbes that grow in oxygen or in the air have the enzyme catalase. But there are a few cells that lack the enzyme catalase, and they can grow in the presence of oxygen because they have the enzyme peroxidase. Peroxidase takes one hydrogen peroxide, combines it with two hydrogen ions to make two water molecules. And as you know, there are two families of microbes that grow in the air and are catalase negative, and that's streptococci and enterococci. And why they can grow in the air is they have peroxidase, so that when hydrogen peroxide is made, they can break it down into water with peroxidase. Any question about that? So our cells have catalase. Most organisms that grow in the air do have catalase. But there are two families that do not, and that's enterococci and streptococci. All right, the fourth toxic form of oxygen is a hydroxyl radical. Once again, a radical is an molecule with an extra unpaired electron, shown by that open square box. Note that hydroxyl radical is different than hydroxide ion, OH with a negative charge. This has two electrons which are paired. And so OH with a negative charge is not damaging to cells. And then OH in alcohols has no unpaired electron. So hydroxyl radical has only one electron in the hydroxyl radical. And then when this bumps into another molecule, it can steal an electron from that other molecule, and that will damage the other molecule. Hydroxyl radical is formed in aerobic respiration in trace amounts. It's also formed in ionizing radiation. So whenever you're receiving ionizing radiation, like with your teeth x-rays, you do make some hydroxyl radicals. And it also is formed whenever hydrogen peroxide reacts with or combines with a certain metal ion. How the body deals with hydroxyl radicals is we have antioxidants which bind to, neutralize, and then remove the hydroxyl radical. Two antioxidants which remove hydroxyl radicals include vitamin C and vitamin E. The trouble is that if the vitamin C does remove the hydroxyl radical, that vitamin C is used up. You cannot recycle it and then use it as vitamin C. So if it's used as an antioxidant, it neutralizes the free radical, and then that molecule is used up. All right, any questions about anything? So we can determine the requirement for oxygen by growing a microbe in a tube that removes oxygen. And aerobes, recall, use oxygen, and they produce more ATP from a molecule of glucose than an anaerobe. We can classify microbes on the basis of the effect oxygen has on their growth. So what you do is you inoculate a thioglycolate tube, shown here. Thioglycolate is a media, or a media component, which combines with and removes oxygen. And so the thioglycolate tube, when you first get it out of the incubator, there will be no oxygen in the tube. But then oxygen will come from the air and start getting in the tube. That will mean that in the tube we'll have complete oxygen, or around 21% at the top of the thioglycolate media. And as you go down the tube, you'll get less and less oxygen. The further down you go, the less oxygen, until you get to the bottom of the tube, which will essentially have no oxygen, because all of the oxygen will be combining with thioglycolate, and then thioglycolate will be removing it. Any question about any of that? So one of the easiest to discuss is an obligate anaerobe. It can only grow at the bottom of a thioglycolate tube. And at the bottom of the tube, it'll have the most growth. As you grow up the tube, you'll have less and less growth. So let me put one dot right there. You'll have less and less growth the higher up you go, because you'll get more and more oxygen the higher up you go. And so you get less and less growth the higher up you'll go in the tube. Eventually, someplace at the top of the tube, you'll get no growth. But in this case, you're getting no growth about halfway down the tube. Come on, mouse. And that's an obligate anaerobe. You also have an obligate aerobe, and these organisms require oxygen for aerobic respiration. So they will grow best at the top of the tube, where they'll have most growth at the top, because that'll be 21% oxygen. And as you go down the tube, you'll get less and less growth. Trying to make an arrow. So you might have like one here, and then one there, it's about the same, and then three right at that level. So what we should do is say two at this level, and three at that level, and then down a little bit more, one, where you do get a little bit of growth because there's a little bit of oxygen. But once that oxygen is used up, the cells cannot grow any further because there's no more oxygen around for the cells to grow. And as you go up the tube, you'll see more and more growth because there will be more and more oxygen until you get at the very top of the tube where you'll see the most growth. And that is an obligate aerobe. Come on, mouse. Both of these differ from a facultative anaerobe, and that is a facultative anaerobe can use aerobic respiration and actually prefers to use aerobic respiration because it can make the most ATP this way. But if oxygen isn't around, the organism can grow because it doesn't require oxygen to grow. It'll just switch from aerobic respiration to some other form of respiration. And that way we can see growth essentially everywhere in the tube, but you'll notice the highest growth at the very top of the tube. And that's because these organisms are growing with oxygen, and so they're growing the fastest. These organisms are falling back on some other way to make ATP other than aerobic respiration, and so they will have less growth. But you will see growth all the way down to the bottom of the tube because some other means besides aerobic respiration is allowing the cell to grow. And those are facultative anaerobes. You have an aerotolerant anaerobe, and it does not require oxygen, but it has all the enzymes it needs to grow in the presence of oxygen. So these organisms can grow everywhere uniformly in the tube. That's different from a facultative anaerobe, which grow best at the very top of the tube. These grow best everywhere in the tube. And those are aerotolerant anaerobes. And then lastly, we have a microaerophile. These organisms are aerophiles, so they must have oxygen in order to grow. However, these organisms grow best at reduced oxygen concentrations. They do not grow best at 21% oxygen or near 0% oxygen. They grow best somewhere in the middle of the tube. And that will be where oxygen is something like 1 to, is it 10% oxygen? And when you go away from the middle of the tube, you can see some growth, but it'll be less growth than the middle of the tube. And then you go extreme, you'll have the least amount of growth. And down here in extreme, you will probably have no growth because there's no oxygen. So the organisms cannot grow at the bottom of the thioglycolate tube. Any question about any of that? All right, I think this is a good place to stop, so I think I'll stop here. Any questions? If not, I'll see you in the lab at 6:30. Goodbye.